The overall goal of this procedure is to harvest O-linked glycans. This is accomplished by first resolving glycoproteins by SDS-PAGE. The second step is to wash gel pieces containing the glycoprotein of interest. Next, the O-linked glycans are released by reductive beta-elimination. The final step is glycan permethylation with phase-partition. Ultimately, permethylated glycans are analyzed by tandem mass spectrometry to detect structures and quantify abundances.
The main advantage of this technique over existing methods, like high-performance liquid chromatography, is that our method separates neutral and charged permethylated glycans in a rapid phase partition.
This method can help answer key questions in the glycoscience field by allowing comprehensive characterization of O-linked glycans released from resolved glycoproteins.
Prior to starting this procedure, resolve glycoproteins using gel electrophoresis. After electrophoresis, place the gel on a glass plate and excise the region of interest using a clean scalpel. To increase the yield of O-glycans, transfer the gel band of interest onto another glass plate and cut the piece into approximately 2 mm cubes. Transfer the small gel pieces into a 13 by 100 mm screw-top glass tube with a stainless steel microspatula. Next, add 1 ml.